Here's part 3 of chapter 9. We were discussing pleiotropy, the ability to change multiple characteristics with one gene. Let's look more closely at sickle cell anemia. So you've got that abnormal hemoglobin, the abnormal shape of the cells. That causes several issues that lead to different homeostatic imbalances, different organ failures that can be fatal. Polygenic inheritance, on the other hand, is multiple genes affecting one characteristic. For example, skin color. So there's a very wide range of pigmentation levels, the amount of melanin that your skin makes, from very light to dark. And that's based on several genes together. Along with our genetics, the environment also plays a role in our phenotype. For example, height and weight can be determined by our nutritional content, as well as our immune response and our genes. Another example would be rickets. That's where children don't have the nutritional requirements of enough calcium and phosphates to put down bone and make it hard. So instead, their bone is softer, and that means that it can bend, and that causes deformation to occur as they grow. It's very sad that this child had all the genetics for proper functioning limbs, but because of malnutrition, they were unable to properly express these traits and build strong bones. Air pollution can cause bronchitis. Our phenotypes are not determined by genetics alone. There's also the idea of epigenetics, where basically, dependent on how genes have been passed on, sometimes they'll be methylated, which means they have a carbon on the outside, and so they can be harder or easier to read. And based on even our grandparents, we can have differential gene expression, which genes are actually expressed and made into proteins, and that can affect our phenotypes as well. In case we haven't hit this hard enough yet, there is a theory called the chromosome theory of inheritance that states that genes are located on chromosomes, and how chromosomes separate during meiosis and come together during fertilization indicates our inheritance pattern. As with everything else that we teach in biology, it's based on the best knowledge we have of our theories. We get some really interesting things happening when we look at the sex chromosomes, the X and Y. So obviously our sex chromosomes determine our sex. And we looked at that there can be different non-disjunctions and different genetics other than XX and XY. It's not just a straight male versus female, but in general, two X's, female, males are XY. They have sperm with a Y chromosome. So it's really the male that determines the sex of the offspring. So back in the day when kings divorced women or beheaded them for not giving them sons, it was the king's fault and not the woman's, because she can only give X's. What's really neat is that in each cell, only one of these X's is active. The other is inactive, and it's what's called a bar body. So males only have one X, females have two, they only have one active in each cell. But males, because they have only one X, are much more likely to get X-linked diseases because they only have one chance at a good allele. If they get a mutated version, they've got those recessive disorders, whereas females have two chances. So let's look at sex chromosomes a little bit more and then the traits that they can influence. So this shows how the male can determine the sex. Females, each time you're going to get 22 normal autosomal chromosomes and an X, whereas males, you can have the 22 chromosomes in either an X or a Y. And this is the same as in humans and fruit flies. Although, fun fact, chickens, XX is a male, XY is a female. Okay. Let's look at some of those X-linked genes and the X-linked recessives that I was talking about before. So you'll have a mother with one normal X chromosome and one that has the altered gene. She's a carrier. The father, he's got a normal Y chromosome and a normal X. There, if the mother has one bad X and she passes it on to her son, because he passed on the Y, there's only one X, you're going to have that son with the affliction. She can have a carrier daughter or unaffected daughters or sons. One example is color blindness. So we have these cone proteins in our eyes that take in color and tell us about it. What you should see here is a 7. If not, you're colorblind. You can't see the red versus green cones. Those light sensitive cells are cones. And unless most people take this test, they won't know that they're colorblind. They just see things a little bit differently. They might have a problem distinguishing between certain colors. And with this X-linked recessive, if a male is colorblind, he had to get that bad copy from his mother. If a female is colorblind, her dad has to be colorblind because she has to have 2 bad copies. Another example would be hemophilia. So this is a disease where you can't properly form blood clots. So blood loss is severe. You saw this a lot back in old royal areas where they had lots of breeding between relatives and cousins were marrying cousins to keep that wealth and land in their own family. And so you saw female carriers, and if they had sons with only a bad X, they wouldn't be able to clot properly. And that would be fatal. Okay, this is the end of chapter 9. I hope you enjoyed genetics.